everyone, I'm Scarlett Li-Williams, the founder and lead of SAAS Missions, and I'll be talking today about the recent scientific experiments my team and I have been conducting in collaboration with the University of Sydney's Payload lab and UNSW student groups and faculty members. So SAAS Missions is a student-led research team based at the Australian Centre of Astrobiology, UNSW, Sydney, and is made up of undergraduate and postgraduate students from universities all over Sydney, including UTS, University of Sydney, Macquarie University and UNSW. Our projects, also known as our missions, include conducting research projects, STEM outreach to students, and workshops that apply knowledge learnt at universities in our projects. Founded in 2018, we have currently 3 active missions, including a mission to the International Space Station, an outreach program on plants in space, and having recently received a grant, creating promotional STEM outreach videos covering topics in astrobiology. For more information about what we do and our missions, please visit our website. It is currently being revamped, but you can still contact us there. So today I'll be focusing on our main mission, hummus, which is named because of our current work with chickpeas. So chickpeas are a type of legume crop, and legumes have a very important role in agriculture and feeding livestock as well as soil health. Other examples of legume crops are alfalfa, clover, beans, peas, lentils, soybeans, and peanuts. So most legumes have a symbiotic relationship with nitrogen-fixing bacteria, such as rhizobium. They grow in these structures in the root called root nodules, as you can see here in this picture. So nitrogen fixation is a biochemical process that takes the atmospheric nitrogen and converts it into a usable nitrogen compound that plants can actually take up and utilize. The bacteria fix the nitrogen required by the plant for multiple functions, specifically growth and increasing their photosynthetic capabilities. In return, the plants provide sugars and carbon compounds for photosynthesis, which the nitrogen-fixing bacteria can then use for energy and necessary processes. This exchange of carbon sources and nitrogen compounds mean that the legume plants and the bacterium can mutually benefit from one another. So in 2019, in collaboration with the University of Sydney's Payload lab, UNSW BlueSat, and the AIAA Student Society team at UNSW, with donations from Biohacking Society and the Burns Lab, UNSW, and sponsorship from UNSW, BABS and B Schools, we created an experiment and a high-powered rocket launch in Queensland to test the effects of high-level G-forces and acceleration on pure strains of rhizobium culture in liquid medium, inoculated peat, and on Petri dishes, as you can see here. So, as seen in this workflow, a 2U unit was built to keep the bacterial payload enclosed and protected, and this payload also housed sensors for temperature and measuring acceleration. This was placed inside the rocket alongside some peat in bags. And we launched it. We also had an identical replica kept on the ground as a control. So once returning to the lab, we took our samples and used the rhizobium cultures from the rocket payload and the ground payload from the plates, peat and Petri dishes to inoculate young chickpea plants, as you can see here. And here is just some of our seedlings growing inside the greenhouse and growing their nice root nodules with our bacterial cultures. So firstly, we found that when comparing the rocket to the ground cultures for the small Petri dishes, there was a statistically significant amount more, over double. In contrast, the liquid medium broth tubes had significantly less in the rocket compared to the ground. In the peat, we found there was no significant difference between the rocket and ground cultures. Both were able to successfully grow nodules in the chickpeas and carry out nitrogen fixation. However, there was an identified potential contaminant that we found. This contaminant showed morphological similarities to rhizobium, so we potentially thought that it could be rhizobium, but it was mutated. However, there's no conclusive evidence to what this strain was, and they were unable to nodulate the chickpeas. So what's next? Our team's next goal is to conduct hummus 2 and grow chickpeas and rhizobium on the International Space Station for 10 days. Our team has been kindly donated a plant growth chamber from collaborators in the US at the International Space University. The chamber being used is the Hydra 1 Plant Growth Chamber and Control Unit, which carried seeds to the International Space Station within this plant growth chamber in the Ice Cube facility. We aim to replicate a similar experiment but modify the chamber to fit our seeds and bacterial cultures. The biological payload that we will use will include a 10-day-old plant and root system with the nodules established. We plan to collect the data on hydrogen production, a nitrogenase assay, and monitors to detect ammonium levels. We also will have standard monitors just to measure the control variables, and we also aim to investigate nodule colour, count and size once returned. We are currently looking for sponsors and supporters for this mission. If you are interested, please get in contact with us via our website or email myself, Scarlett, at scarlett.li-williams@hotmail.com or s.lee-williams@unsw.edu.au. Thank you very much for listening to this brief presentation, and please feel free to reach out with any questions you have on the experiment that we've done or plan to do. You can ask this via the Slack channel or just send me an email.